Okay, so good day everyone. So today we will be discussing the different hemolytic patterns. So ang hemolytic patterns natin is significant for enterococci and streptococci bacteria. Okay? So So there are numerous ways to group and identify these two medically important species or these two families of bacteria. So initial identification of these bacteria is often based sa hemolytic pattern nila sa blood agar plate. Okay, so some species ng streptococci and enterococci, they produce exotoxins that completely destroy red cells in the blood agar plate. Kaya yun yung nagkakos ng hemolytic patterns. So after patterns of hemolysis have been identified, species of streptococci and enterococci can be further differentiated based on their cell-borne carbohydrates. Okay. So this method is developed by Rebecca Lansfield in the year 1930, which uses an alphabetic system called the Lansfield serological group. So, ano nga ba yung hemolysis na tinutukoy natin? This is the destruction of the red blood cells on the back culture media. Okay? This is due to exotoxins or virulence factors that are produced by streptococci and similar organisms. So, blood agar medium is a solid growth medium that contains red blood cells. Okay? So the medium is used to detect bacteria that produces the enzymes to break apart the blood cells. So blood agar medium is also a type of a differential medium, okay, made up of 5% sheep red blood cells. Okay. So rabbit or horse blood may also be used for growth of NAD requiring organisms such as Haemophilus species. Okay, but the hemolytic pattern may be inconsistent with those na sheep blood yung gamit. So the blood used in agar is also treated beforehand to remove a molecule called the fibrin. Okay? Fibrin participates sa clotting ng ating blood, di ba? Okay? So the absence of fibrin ensures that clotting of the blood does not occur in the agar. So, yung fibrin kasi possible na makapag-interfere sa hemolytic reaction. Okay? Also, human blood is discouraged to be used para sa safety purposes din. Para maiwasan natin yung exposure sa bloodborne pathogens such as HIV or hepatitis. So BAP tests the ability of an organism to produce hemolysins. Okay? Hemolysin is yung enzyme na tinutukoy natin, the exotoxin that breaks apart red blood cells. Okay? So the degree of hemolysis by this hemolysin is helpful in differentiating members of the genera Staphylococcus, Streptococcus, and Enterococcus. So we have different types of hemolysis. Okay, so the determination of hemolysis and of the hemolytic reactions is useful in distinguishing different types of bacteria. Okay. Subsequent biochemical testing can narrow down the identification even further. Okay. So, tinitignan natin yung hemolytic pattern niya to give us a vague idea kung anong family or type of bacteria ba siya. And then, doon tayo magpo-proceed sa biochemical testing. So we have four types of hemolysis. We have the alpha, beta, alpha prime, and the gamma. Okay. So to read the hemolytic reaction on a blood agar plate, the plate must be held up to a light source and observed with the light coming from behind. Let's start off with alpha hemolysis. Okay. Alpha hemolysis is it shows partial lysis of red blood cells around the colony. Okay. So take note of the greenish discoloration of the area around the colony. Greenish or kind of brownish discoloration. So alpha hemolysis is the reduction of the red blood cell hemoglobin to methemoglobin in the medium surrounding the colony. So this causes a green or brown discoloration in the medium. Okay, again, yung hemoglobin nagiging methemoglobin. Kaya siya nagkakaroon ng greenish or brown discoloration. Example ng mga groups na merong alpha hemolysis, we have the S. bovis group. S. bovis group is they are non-enterococcus members of the viridan streptococci. We have A. faecalis, Il falciparum, they are member of the enterococcus. You have your S. pneumoniae, S. pneumoniae is important sa identification ng pathological bacterial pneumonia. Okay, pathologic cause ng bacterial pneumonia and meningitis. S. pneumoniae. You have anginosus group, mutans group, the metis group, salivarius group. They are members of the viridans group. They are mostly normal flora and not pathogenic. Okay, so some groups exhibit weak alpha hemolysis and a few alpha hemolytic types may exhibit variability. Okay? So, nagbabago-bago. Yun, a few groups ng bacteria natin. Mamaya may mention ulit yun. Okay. Let's give, i-note natin to si Streptococcus pneumoniae. This is a significant human pathogen, again, causative agent ng bacterial pneumonia, and can cause meningitis din siya. Also, causative agent ng otitis media or ear infection sa mga bata. Streptococcus pneumoniae does not possess a Lancefield antigen. Blood agar colonies niya, they are smooth, mucoid, and surrounded by a zone of greenish discoloration. So, yun nga yung ating alpha hemolysis. So, presumptive identification of S. pneumoniae can be made with a positive off-the-chain susceptibility test. So, paano po yun? If you have seen an alpha reaction or alpha hemolytic pattern sa blood agar plate, you could suspect na it's under possible Streptococcus pneumoniae. Okay. So you can further confirm by using off-the-chain susceptibility test. Okay. Pag nag-positive ka doon, so you can say talaga na it is Streptococcus pneumoniae. So, next, let's proceed with the beta hemolysis. Okay. Beta naman, this is the complete lysis of red blood cells around the colony. Okay, so complete lysis na siya. There is clearing around the area of the colony. Yan. So, this is yung colony. Ito yung area niya. Yan, nag-clear na siya. So, beta hemolysis is caused by two hemolysins, that is hemolysin O and hemolysin S. Okay? So, hemolysin O is inactive in the presence of oxygen. So, thus, yung stabbing of the plate, stab strict technique ang ginagamit natin. Okay? Yung stabbing increases the hemolysis reaction. Ay, yung stabbing kasi, ang ginagawa niya is ine-expose niya yung agar sa air. Okay? Kasi nga yung hemolysin O, sorry. Yung hemolysin O is inactive in the presence of oxygen. So, ini i-stab mo siya, so malalim yung parang malalim yung, or in between siya ng agar, di ba? So, hindi siya masyadong exposed sa hangin. Okay? So, yun. Para mas mag-react si hemolysin O. Okay? So, S naman, kabaliktaran, hemolysin S is oxygen stable naman. It is an oxygen stable cytotoxin. Okay? So, for beta hemolytic bacteria, ang recommended talaga is stab streak technique. Okay? Stab the agar plate several times para sa oxygen-sensitive na hemolysin, which is the hemolysin O. Ayan. Ganyan yung stabbing. Okay, so itutusok talaga siya. Hindi siya kakayurin. Hindi kakayurin yung agar. Itutusok lang talaga yung loop. Okay, so stab technique. You have the group A streptococci. Okay, let's discuss yung eto muna. Ang example ng mga may beta hemolysis, you have your S. pyogenes, S. agalactiae, S. dysagalactiae, S. equi, aginosus group, mutants group, mitis group, salivarius group. They show variability, kaya possible beta hemolysis din ang show nila. So, let's discuss further yung different groups. We have first your group A streptococci. Group A streptococci, ang main representative here is the Streptococcus pyogenes. Streptococcus pyogenes. So, in order to differentiate S. pyogenes from other streptococci and enterococci of this group, okay, so isolates are tested for resistance to bacitracin. For example, a beta hemolytic reaction is indicative of a streptococcus, diba? So beta hemolytic yung nakita mo, so may isip mo na agad streptococcus. So how would you know the specific species? Okay, you can test for bacitracin resistance. Okay, so testing of the streptococcus organism with bacitracin is often done to further differentiate them. So, bacitracin is an antimicrobial that is produced by the bacterium Bacillus subtilis. Okay, Streptococcus pyogenes strains are almost uniformly sensitive to bacitracin, but other antigenic groups of Streptococcus are not bacitracin sensitive, meaning they are resistant. Okay? So, if you have tested your known samples sa bacitracin and nag-react siya, that is most likely Streptococcus pyogenes. Kasi again, other Streptococcus species are not sensitive to bacitracin, meaning they are resistant. So, walang reaction. Okay? So, si S. pyogenes ang sensitive kay bacitracin. Next, you have your group B, streptococci. So, ang main significant natin ditong species, you have your Streptococcus agalactiae. Okay. So, this is the only recognized species of the Lancefield group B. So, preliminary identification of this species relies heavily on a positive camp reaction. So, we will discuss this further later on, yung camp reaction. Next, we have Group C. Group C, most important naman here, you have the S. dysagalactiae. It is the most clinically important in this group. Okay? They are seen in patients with underlying illnesses. So, uncommon siya na human pathogen but may be involved in zoonosis. Presumptive identification for this agalactiae is from other beta hemolytic streptococci. So, is based primarily sa resistance ng bacitracin and a negative camp test. Okay? Si Group A is sensitive to bacitracin. Si Group B is positive sa camp test. So, how would you differentiate si Group C? Group C naman, kabaliktaran ng dalawa, it is resistant Okay, bacitracin and negative ang CAMP test. Okay. Next, we have a type of hemolytic pattern. You have your alpha prime. Alpha prime naman is a small area. Meron siyang small area of intact RBCs around the colony surrounded by a wider zone of complete hemolysis. So, meron siyang parang fish eye. Ito, tignan niyo yung cursor, this one. So, parang ganyan siya. Yung colony nasa gitna, may RBCs around, small area lang. And then, a greater wide area na complete yung hemolysis. May picture ata dito. Later on. Okay. Next, we have gamma hemolysis. Gamma hemolysis naman is yung no hemolytic pattern. So, walang RBC na lyse. Okay, walang change sa agar. Okay. So, organisms na may gamma hemolytic pattern, they do not produce hemolyzing enzymes that are essential for the breakdown of red blood cells. Okay. So, most notable species here, we have the Enterococcus faecalis. Okay. Enterococcus faecalis is formerly called Group D. Group D. Okay. So other streptococci included in this group are usually not pathogenic. So you have lastly group B. Group B is sa Enterococcaceae family. Ang group B and are considered variably hemolytic. Okay? So variably hemolytic, ibig sabihin they can show a hemolytic pattern or they can or not or nag-iiba-iba siya minsan. So, in Rococca group, we have pwede dito si A. faecalis and A. Faecium. So, in blood agar, enterococci form large colonies that can appear non-hemolytic, alpha-hemolytic, or rarely beta-hemolytic. So, isolates that are able to grow in the presence of 6.5% NaCl and are able to hydrolyze bile esculin are presumed to be enterococci in this exercise. Okay? So, if those colonies na nag-react possibly non-hemolytic or alpha-hemolytic, pwede natin silang i-test sa 6.5 NaCl and bile esculin. So, pwede, para ma-differentiate natin sila further. If they reacted or may reaction with these two, we can say that they are from the group D enterococci. Okay. Sample inoculation natin or sample procedure, for example. Okay. For example, diretso na tayo here. Ayan. For example, magkakuha tayo ng sample from pharynx ni patient. So, we can do directly na galing sa pharynx or we can use a unknown mixture or broth. Okay? If you're gonna be taking sample directly from your patient, you can use a cotton swab. So, isaswab po directly sa pharynx. Sorry. Okay, you can take directly sa patient sa pharynx and then tsaka siya isaswab. Yung cotton swab na yung gagamitin natin for this part. So, isaswab lang to here sa part na to. Okay? So, if you're gonna be using unknown mixture naman or ng broth, so same pa rin, kukuha ka kay patient, kukuha ka ng sample from the pharynx, and then yung cotton swab, dito mo siya ilalagay sa inyong broth. Yung broth now, dun ka naman ngayon kukuha ng sample na isa-swab mo rin on the same manner. Okay? So, isa-swab yung here. So, dito. And then, four-quadrant technique na para makakuha ka ng isolate colonies. Okay? So, incubate at 37 degrees Celsius for 24 hours. Kapag meron ng tubo, depending on the hemolytic pattern, tsaka natin malalaman kung ano yung next step nyong biochemical test. If alpha hemolytic yung pattern, so we can subculture it sa trypticase soy broth. Okay? And then, same with beta hemolytic pattern, subculture din with trypticase soy broth. After the subculture, ayan, proceed with biochemical testing. Yung alpha hemolytic pattern yung naging reaction niya. So, iti-test natin for optogene susceptibility test, sa bile esculin, and sa 6.5 NaCl. Okay, to further differentiate them. Sa, ano naman, for the beta hemolytic, iti-test natin for bacitracin, okay, SXT, and CAMP test. So, all media is incubated at 37 degrees Celsius for 24 hours. So, ito yung ating streaking technique. Okay. Firstly, yung swab is rolled over approximately 1/5 of the area. So, yung cotton swab, dinidip lang here or ini-swab siya directly here. Once okay na yan, get your loop, tapos iti-thin out na natin siya using a 4-quadrant streaking technique. So, ayan. First quadrant, second, third, and then fourth. And then yung stab ninyo is dun sa areas na walang streak. Dun nyo siya isa-stab. So, stab them a few times. Iwasan ulit yung area na na-streakan nyo na. So, let's look at this para mas ma-differentiate nyo pa yung hemolytic pattern. So, for beta again is complete lysis. For example, this is the colony. So, ito yung zone niya. So, wala na talaga natirang RBCs. Kung titingnan nyo yung sa stab, so here is the step growth, and then sa paligid niya wala ng RBCs. For the alpha prime, ayan, para siyang fish eye, you have your colony, tapos thin layer ng RBCs, and then complete lysis sa outer layer. Okay? And then for alpha is partial hemolysis lang. So, meron pa rin konting natira. Okay? Ayan. Tapos, wait lang. Namiss ko ata yung camp test. Saan nga ba yun? Here pala yung camp test. Yung camp test. Okay, let's discuss yung camp test. So, hemolytic reactions they can display some synergy. Okay? For example, si Group B Streptococcus agalactiae, they are weakly beta hemolytic lang. Weakly beta hemolytic. However, in presence of another strain ng Staphylococci, okay, so it can react more intensely. Okay, so the beta hemolysin of the two organisms can combine to produce an intense beta hemolytic reaction. Okay, so nagko-combine yung reaction nila to produce a stronger reaction. So, ayun yung, pag ganun yung nangyari, that is what we call positive camp test. Okay? Parang confirmatory test siya for beta hemolysis. Okay? So, kasi since weakly reacting lang siya, sometimes hindi siya nakikita. Hindi nakikita yung reaction. So, to confirm if gamma ba talaga, walang reaction, or weakly lang siya, we do CAM test. So, ihalo natin yung inyong, hahaluan nyo yung colonies or yung plate ninyo ng another strain, which in this case is Staphylococcus. Then, if they reacted or nagpakita na si beta hemolysis, ibig sabihin, positive siya for CAM test. Ayun, nalagtawan pala yun kanina. And then, ayun, lastly, here are sample photos. So, give the type of hemolysis. Let's start with C. As you can see, walang nangyayari sa agar. Kita lang yung streak niya, pero hindi walang discoloration or anything. This is gamma. B is, nakikita niyo greenish discoloration. May yellow part or nag-clear part. Konti lang, pero mostly is greenish. This is alpha. And then sa A, kita niyo, wala na talaga siyang RBCs doon sa mga part na may streak. So, nag-clear, totally clear or totally nag-lyse na yung RBC. So, that is alpha. Beta. Beta hemolysis, complete lysis. Yeah. So, that is all for this discussion. If you have any questions po, feel free to message me na lang. So, thank you po.